All right, sorry about that. My computer's going very, very slow today. So, good morning. Today we're talking about translation, which is the second part of protein synthesis. It's a little bit confusing because now we're translating all of that information into amino acids so that way we can make them into proteins. So today we're just talking about translation, the second part of protein synthesis, and then you do have an assignment on translation. So remember, protein synthesis is the making of proteins. Synthesis means to make, so yes, protein synthesis means to make proteins. We talked about the very first step, transcription, yesterday. That happens inside the nucleus with the DNA changing into mRNA. Well, now we're talking about step 2, which is translation. We're going to change mRNA into a protein. So just to review step 1, transcription. During transcription, mRNA is copied from a single gene that codes for a single protein. Remember, we're not copying the whole DNA strand. We're just going to copy the part we need to get that protein, to get that job done. We don't need all of it. We just need some of the DNA. The DNA is inside of the nucleus. The mRNA carries the instructions or the code for making the proteins. And when it leaves the nucleus, it finds a ribosome in the cytoplasm, and that's how we get to step 2. So now we're on step 2, translation. Translation is the process of making a protein from the mRNA code, which again, protein synthesis, that's what it makes, making protein. So translation decodes mRNA's message into a protein. So we're going to learn how to take 3 digits and make them into an amino acid to make them into a protein. It does take place on a ribosome in the cytoplasm, and that ribosome is known as RNA. So again, mRNA leaves the nucleus into the cytoplasm where it finds a ribosome, and this is where all the translation occurs. So before we start talking about the steps, I want to talk about some keywords, codons and anticodons. So the mRNA strand itself, the one that leaves the nucleus, has a codon on it. And a codon basically is just a group of 3 nitrogen bases. We're going to group them in 3s because it's easier to code based on 3s. So codons are on the mRNA. They come in 3s, all 3s. An anticodon, that's going to be located on the tRNA, which is one of the ribosomes, one of the RNAs that we read about. Today we're going to explain it. But a tRNA is going to carry the anticodon, and basically it's just a match to the RNA. For example, right here we have AGU. Remember, RNA doesn't have a T, so there's no T's. But A goes to U, G goes to C, and U goes to A. That's how it matches up. Again, the codon's on the mRNA, and the anticodon is on the tRNA. So these are the steps of translation. First, again, the mRNA that left the nucleus is going to attach to a ribosome called rRNA. And it's going to attach at the start codon, and the start codon is very, very important. Translation will not begin until the start codon AUG is read. That is the only set of 3 letters that will begin the translation process. So that ribosome is going to read the mRNA until it sees the letters AUG. Once it sees those letters, then it's going to start making protein. It will not make the protein until it sees those letters AUG. That's the start of everything. The transfer RNA, or the tRNA molecule with the correct anticodon, is going to attach to the mRNA inside the ribosome, and with it, it's going to bring an amino acid. So The mRNA, again, finds the ribosome. The ribosome's going to read it until it sees those 3 letters, AUG, which is the start codon. Then we have the tRNA that's coming inside. It's coming inside the ribosome, and it's only going to match depending on if the anticodon matches the codon. So AUG, again, matches with UAC. UUC matches with AAG, so on and so forth. With it, it's bringing this amino acid. And if you forgot, amino acids are monomers of proteins. So amino acids all together are going to make the protein strand that it needs. And it's just going to bring it in one by one. So this protein, this tRNA had a protein. It was the green one, but it attached it to the new tRNA. And then these 2 are going to leave and attach to the red. Once that dark blue gets in, those 3 are going to detach, and then it's going to attach to the blue. And it's just going to keep going and going and going. All the tRNAs are going to leave when they don't have an amino acid anymore. They're going to go out, get another one, so they could come back and do this process all over again. Then the mRNA is going to slide down 1 codon at a time. So each set of 3, it's going to just like an assembly line. 3 goes in, 3 goes out. 3 goes in, 3 goes out. And that tRNA is going to bring in the next amino acid, just like an assembly line. Amino acid in, tRNA out. Amino acid in, tRNA out. Amino acids are going to join by forming peptide bonds, and that's what's in that picture right here. Connecting the amino acids, you see a straight line. That's the peptide bond. This continues until a stop codon is reached and amino acid strand is released. So there's more than one stop codon, and we're going to see that later when we look at the chart. Then amino acid's going to fold into a protein. So it looks like a long chain at first, and then it's going to fold into a globular blob, basically. So this is the amino acid code, and this is how we're going to translate things into a code. Again, on mRNA, we have codons. Those are 3 sets of nucleotides. Each codon on the mRNA is going to code for amino acid, and this is the chart we're going to use to code for that. Because inside the body, our cells are using a chart to code as well. It's a little complicated, but once I tell you how to figure it out, it'll all be easy, I promise. The first thing you're going to do is you're going to use the first base on the left. So whatever your first letter is, you're going to pick that row, U, C, A, G. Then your second base is on the top. So if your second letter, you're going to pick that column. And then your third base is on the inside. So first letter, second letter, third letter will tell you which one it is. And then these funky letters next to it, that's your amino acid in a shorthand form. They're really long names, but we just write them short for the sake of the codon chart. All right, so let's go over some examples so that way I can make sure you understand what we're doing. Actually, we're not doing an example yet. No, I can give you an example. All right, ready? The first thing I'm going to tell you to find is going to be a G, a A, and a U. G, A, U. Let's see. And I just made those up randomly. G, A, U. So first letter is G. Second letter is A. So that means we're picking everything in this row. And then since we're A, we're only picking from this column. And then the last letter was U, G-A-U. Our amino acid is ASP. Make sense? Any questions? Let's see if you can try it out. Open up your chat room for me. Set it to private. Don't tell anyone the answer. Let me clear this out. I'm going to give you a set of letters. Let's go with AUC. So go ahead and find me the amino acid for AUC. Whoops. How do you get rid of that? Oh, well. AUC. Find it for AUC. Whoops. Oh my God, I'm freaking out over here. Good job, good job. So everyone so far that sent it to me in the group chat got it right. Good job, guys. All right, let me give you another one just to be safe. How do I get rid of something that I already typed? Erase it. Ah, there we go. All right. I'll give you the letters C, A, G. C-A-G. C-A-G. Good job, good job, good job. You get it, Wyatt? You got it? Okay. Good. All right. And if you were here, we would play bingo, and I would give you all these letters, and I'd give you candy if you won. But we don't have school anymore. And it's really hard to play bingo on a computer. All right, so this is another anticodon chart. We don't really use this one too much, but it does exist. Same principle. I actually think this one's easier to use. So the very first thing you're going to do is you're going to start in the center and work your way around. So let's say my letters were G, C, A. Well, you're starting in the center, so you start with your first letter G. So that means all this crosses out. You don't even need that. Then your second letter C, so you're going to go to this section. You don't pay attention to any of this. And then your last letter is A. So A would tell you it's alanine. That's why I said I actually think this one's a lot easier to use. But we have to show you the other one because we have to. All right, so I'm going to give you another example for you to find. Let's go with the letters C, U, G. C, U, G. C, U, G. Good job, good job. C-U-G. Yes, good job. So yes, C-U-G. Again, you start with the first letter, C, second letter U, last letter G. All right, let's give you another one. A-U-G. A-U-G. Let me erase this real quick. A-U-G. Good job, good job. We're doing great. Yes, good job, everybody. So again, A, U, G. So this codon chart isn't too hard. It's pretty easy. Hopefully, makes sense to you. All right, so let's put it all together, starting with the DNA. We're going to transcribe it to mRNA, and then we're going to translate it into a protein. All right, so this is our original DNA strand. We want the mRNA, and then we want it broken into codons. So I am going to underline, well, circle, I guess. I already started circling 3 at a time. I am terrible with this mouse. I feel like I did something wrong somewhere. Oh, thank you. Oh my goodness, let's just erase that part. Ah. Okay, so we're not making a DNA strand, we're making an mRNA. And on mRNA, remember there's no T's. We use U's instead of T's. So let's see. My first T is gonna go with an A. My next A is gonna go with a U. My C is gonna go with a G. Next, I have my A, that's gonna go with a U. C is gonna go with a G. My other C is gonna go with a G. And then my next G is gonna go with a C. Then I have another C because I have another G. T is gonna go with A. Then I got my CCA. That's gonna be G, G, U. Then I have my TTT. That's gonna be A, A, A. Then I have my ACT. A is gonna go with U. C is gonna go with G. And T is gonna go with A. And now I'm gonna find the proteins being produced. So first letter's A. I'm in this row. Second letter's U. I'm in this box right here. And my third letter's G. So I'm either met or start. But since we always start with the first anticodon, this is gonna be our start. Some ugly handwriting. UGG is next. So U, I'm in this row. My second letter is G, so I'm in this box. And my last letter is G, so I'm TRP. Then I have CCA, so C, I'm in this row. Second letter is C, I'm in this box. Last letter is A, so I'm pro. Next, I have a G. So I'm in this one. Then I have another G. Then I have a U, so that's gly. Then I have AAA, so my first letter is A. Box is A. Last letter is A, I'm LYS. Then I have UGA. First letter is U. then I have a G, then I have an A, and that is stop. So this is typically what an mRNA is going to look like when it's making the proteins, except longer. It's going to have a start, it's going to have a stop, but it's going to have way more codons in the middle of it. And yeah, that's how we translate mRNA into proteins. Oh, I need to erase this. Oh, sorry, we don't need this one. We already did transcription. And that's it. So in this last step, we use tRNA and rRNA. In the very first step that we talked about yesterday, we used mRNA. So that's all I needed to talk about today. You are free to go do today's assignment. If you have any questions, stay on. If not, you are free to go.